Our first speaker for today is Emily Mayer from the DEP. She's going to be talking about HAB testing and related topics. So if you would please, I'm going to turn over the microphone to Emily.
Thank you so much for that introduction. So today I'll be talking about NJDEP's HAB strategy. So in 2019, Governor Murphy announced funding for harmful algal blooms. In 2021, NJDEP released its harmful algal bloom strategy plan outlining the primary goals, tools, outreach, and other pertinent information. The primary focus of the harmful algal bloom strategy is a statewide approach to HABs, focusing on all water resources, from drinking water to recreational, streams, rivers, etc. HABs can produce toxins, and this can be potentially dangerous to humans, health, and animals, and this strategy was implemented to help protect them. And so the plan further describes various entities who are responsible for response and actions through DEP, coming through various divisions and external partners who make this strategy possible, as well as detailed information on the parameters and our rapid response and detection strategy. So we'll kind of go over today the tools that NJDEP utilizes and kind of what that looks like, as well as adding new advisories such as our winter advisory to protect public health. And so just a general overview for those who aren't as familiar with HABs. So the NJDEP strategy focuses on cyanobacteria, which is also known as blue-green algae. So cyanobacteria are an ancient microbial life form, which is thought to be responsible for oxygenating the earth over 2 to 4 billion years ago. So cyanobacteria can be found in a variety of environments, and they are a true bacteria. So even though they do act like algae, they're considered bacteria essentially. And they can actually be present in tidal systems as well. So you can kind of see up at the top here, we have a couple of picture examples of what that cyanobacteria looks like under a microscope. And so the cyanobacteria can create something called aconites, which are kind of like spore-like dormant cells, which allow them to survive during adverse and unfavorable conditions. And you can also see kind of that egg-shaped looking cell up at the top. So that's called a heterocyst, which provides the filament with organic nitrogen compounds. So it allows it to fixate on those nutrients in the water column in order to continue to thrive and reproduce. And so, as mentioned, harmful algal blooms can produce toxins that can cause a variety of different health risks. So it can cause allergic-like reactions, such as asthma, eczema. It can also cause different flu-like symptoms. So it's important to kind of look out for our advisories and signs for harmful algal blooms this summer, and also to be vigilant upon letting your pets go and swim, as there have been numerous cases across the country of potential seizures and deaths occurring within animals. And so some of the various drivers and impacts of harmful algal blooms. So weather can be a contributing factor to the formation of HABs through various mechanisms. So through nutrient loading, increased turbidity, changes in water chemistry, transport of algal cells, and stratification disruption. So nutrient loading can occur during rain events, which may carry phosphorus and nitrogen into those water bodies, which can stimulate the rapid proliferation of harmful algal blooms. So you can kind of see in this graphic, there's an arrow pointing down to the sediment that shows cells settling. So some of those aconites or heterocysts that are produced by cyanobacteria can actually stay dormant in the sediment and overwinter. And then when the lakes start to turn over and that stratification or mixing throughout the water column can occur, those cells can resuspend and kind of wake those cells back up, potentially causing HABs. And so these are some photos of examples of harmful algal blooms. So you can kind of see over on the left side of the slide here, we kind of have some floating filamentous algae that can kind of create these mats or cotton-like mats. A lot of times you'll start to see that dying back and start to turn like a brown color, so those cells might start to die. We kind of have a pea soup appearance with some of these pictures as well. You can kind of see in the picture in the middle here, the harmful algal blooms can kind of be wind blown into certain areas throughout the lake. So you can kind of see in the open area, you don't see much of a HAB, but you can kind of see it concentrated behind those lilies. And then on the rare occasion, you might see HABs appear in this picture in the lower right-hand corner, where it looks more of a white or brown look. So when cyanobacteria starts to die back, they can excrete something called lipids. So it kind of looks almost like that oily sheen on the surface. So a variety of different appearances here with HABs. And so these are a few pictures of what HABs don't look like, but sometimes it can be tough to see from the pictures as well until you get on site and kind of do some visual verification. So that's something that we also include in our HAB response strategy. So a few of these pictures are pictures of tree pollen accumulating on the surface. It is spring, our trees are blooming, so I do see potentially an increase of falls coming in soon. And then we also have some duckweed and watermeal that can kind of create these mats as well. So part of our strategy is doing enumeration and identification. So once a HAB sample comes in, it comes into the lab, and I am one of the people that counts all these cells under the microscope. So we only look at cyanobacteria. We don't necessarily count all the other algae within that sample. And so kind of imagine a string of pearls. We count every single circle in that string of pearls, just as an example of how tedious this can be sometimes when you have hundreds of thousands of cells under the scope. And so we calculate those counts out to cells per ml. And so this is just an example kind of showing what blooms can kind of look like, a variety of different assemblages. And so early in the spring, you're going to likely have a mixed variety of different cyanobacteria and algae. So you'd likely see right around this time your diatoms, your green algae, and maybe a sprinkle here and there of cyanobacteria that wouldn't necessarily be at problematic levels at this time. But as the season continues, we'll see increased warmer weather and an increased shift into transitioning into cyanobacteria and green algae. And so we also measure for toxins as well in our lab. So we use a test called the Elijah method. So this is one of our fancy machines. It basically does a whole lot of pipetting. So this is an EPA-approved method through the EPA 546 method for toxin analysis. So we measure for microcystins, which is produced from microcystis, anatoxin, cylindrospermopsin, and saxitoxin. Primarily, we right away, once a sample comes into our lab, we screen for microcystin because it is the most common toxin that we do see. However, we do screen for additional toxins based on what we see in the assemblage, because certain cyanobacteria produce certain toxins. And so here is just a quick table outlining some specific health effects based on certain specific cyanotoxins and what those health effects kind of look like and what specific cyanobacteria species produce those toxins. So I'm not going to go through that entire table, but that's just a quick look there. So as part of the strategy, we do have guidance values that we follow and recommendations that we provide. So these are produced from our alert system, which I'll get to in a minute. But these are the various recreational levels based on each individual toxin. With the Bureau of Freshwater and Biological Monitoring, we don't necessarily have the jurisdiction to shut down a beach. That is with New Jersey Department of Health handles that. So that's why we have recreational guidance values here. And so NJDEP formed our harmful algal bloom expert team. So this is comprised of consultants, academia, researchers. So their primary goal is to provide us with different guidance on HAB prevention, mitigation, and look into various treatments of harmful algal blooms and review data and water quality as well. So most recently, if some of you attended our webinar from Tuesday, which was our lake management guidance webinar, we talked a little bit about our harmful algal bloom lake management guidance that was developed by the HAB expert team. So please check that out online as it is available and has a lot of useful information for lake associations. So we do have on our website a HAB reporting app. So there's a variety of ways that you can report a HAB through our 1-800-WARN-DEP. You can also email us. Also, if you're part of a volunteer monitoring group, you can also report that way as well. And so when you do report a HAB, it goes through survey 123, so you can kind of see there's a picture up there. And when you submit that information, you'll submit pictures as well as various measurements for temperature, phycocyanin, which we'll talk about in a little bit how you would go about doing that. And then once we get that report, it comes into DEP, and then we start investigating further. Our HAB coordinator then will look at the report, and then we'll go about prioritizing what needs to be monitored based on risk to public health. And so, as mentioned before, all divisions are working together, so from Fish and Wildlife to local health departments, nonprofits, state parks, and more. So lots of people involved and lots of partners involved, and we appreciate everybody's help. And so on our website, you'll see there's something called the HAB dashboard. So this is live when a report comes in. It'll pop up on our map here, and you can actually click on one of those dots to kind of see what the status level is, what the alert tier is, what the cell counts look like, and what the toxin results are. And so we have a lot of different tools in our toolbox, screening for the presence of cyanobacteria. So we have continuous monitoring buoys that monitor every 15 minutes for phycocyanin, which is a special pigment that is emitted from cyanobacteria. It's specific to cyanobacteria, so we look at that as a way to kind of screen for HABs. We look at cyanobacteria under a compound microscope. As mentioned earlier, we use the ELISA method for toxin analysis. And we actually do flyovers through the state of New Jersey, I believe, every 2 weeks. So we can screen for HABs that way, and if we see a lake that's looking really green, then, rapid response on that. And so this is our tiered alert system. So some of you may be familiar with seeing our signs out at the lakes throughout New Jersey. But our first alert, called a watch, starts around 20 cells per ml. So that's kind of when we start to get concerned and need to kind of keep an eye on that. And then from there, those numbers increase and start to shift more focused on what the toxin results kind of look like. So again, these are recommendations on the side as far as for public bathing beaches as well. And so this is an example of a few of our signs that we have here. And if you look closer at the signs, there were some questions when these signs originally came out of, well, can I swim? Can I go kayaking? Whatnot. So we added some imagery there to kind of show check mark, yes you can, or a little exclamation point of like with use caution, or you know, the ones with X's, do not do that particular activity. We added our winter watch sign in 2021 because unfortunately HABs are persisting throughout the winter with our warming climate. And so we have a phycocyanin meter loan program. So partners can submit their data to the HAB app. We do have some volunteer monitoring organizations throughout the state that religiously go out every 2 weeks and do their own monitoring internally. And then they decide to share that data with us. And it also helps as an additional screening tool for the occurrence of HABs in general. So our partners are given a standard operating procedure guide to help support them in the use of the meter with lots of useful videos and whatnot. So if you email that email there, again, we have a limited number of phycocyanin meters, which looks like this, on our in our department. So please feel free, and we'll do our best to try to get you one if we have one available. And so this is a comparative summary from 2022 to 2023 of our cell counts and correlations between our phycocyanin meter readings. So a lot of times we get questions of, okay, well, what number should we be looking at on our phycocyanin meter that would alert us to potentially a watch event happening? So we reevaluated our data, and if you start to see around 13 to 14 show up on the phycocyanin meter, then it's a possible watch alert. We have to collect a sample and do further analysis to determine if that is the case. And then again, if you get any higher numbers, so in the 20s, then it could be potentially an advisory alert. So now very quickly, I'm going to go over some of our results from 2023 from last year. So we are reevaluating our HAB strategy again, as we do every year, so that is in progress. And the final 2023 HAB report is currently in review and will eventually be posted on our website. So just last year, we had 88 water bodies that were reported, but we had over 300 reports coming in. So again, this number comes from the public reporting app. We don't necessarily include all the phone calls and additional emails that we get as well, so this is an estimated number. So when we went out to these water bodies, we grabbed around 79 grab samples, or 79 water bodies were visited, and we grabbed samples from there. And then once we take those samples, we pull samples from the samples, so that ends up being over 470 individual samples collected to date. Over 470 samples were analyzed through identification and enumeration, and we performed over 630 toxin analyses. And so this is just an event summary, just kind of showing everyone how many reported water bodies were reported over the years, and then how many of those were actually confirmed HABs once we got on site and collected those samples. So last year it was 49. Water bodies with HAB closures at bathing beaches were 9, and 8 drinking water resources were investigated as well. And so here's a distribution map of the water bodies that were investigated for HABs with the highest alert recorded. So we kind of see it a little bit more concentrated in the northwestern part of New Jersey. So we had 49 water bodies that were confirmed halves, so that was either a watch, an advisory, or a warning alert. 2 of them had the highest alert of warning, 36, so the majority of them were an advisory alert, and then about 11 of them were watch alerts. And the first confirmed halve was at Greenwood Lake at the end of April. And so here we kind of tried to measure out our halve percent occurrence over time. Not all the arrow bars are there because there's a lot of information for that graph to generate. So we had about a 1% increase or decrease in reported water bodies, and then we had about a 28% decrease in confirmed halves. However, because of the weather patterns being very similar to the year of 2019, we suspect that this year might be a bad halve year. So you can kind of see on the graph or the map on the left, over the last 365 day average precipitation events, different areas that could be at higher risk of halves potentially. So the areas that are in blue had a 26% to 50% above average amount of rain that occurred in 2023. And then the areas that are highlighted in green had about 11% to 25% above average of precipitation. Leading into spring, we had a very warm spring leading in, but then kind of cooler temperatures in the early summer months of May and June, peaked again in July, and then again a decrease in August, and then again we went back into a pattern of a warmer winter. So it'll be interesting to see what that graph looks like next year. And so the majority of our samples that were collected were in Morris County. Keep in mind that this is likely because there are larger lakes in Morris County, such as Lake Hopatcong, so there were quite a lot of visits that were occurring as well. So this is just kind of the breakdown county-wise. That can also help us determine where those halves are happening. And so this is the percent of samples collected for screening for microsystems. So you can see that the majority, or 84%, had less than 2 micrograms per liter of the toxin. So for the majority of the samples, had very low levels of that. And then we had about 16% of the samples were 2 to 8 micrograms per liter higher. And so here we're looking at the maximum cell count per confirmed halve samples collected in 2023. Again, 63% of the samples that we received were less than 20,000 cells per ml, so below our watch alert. And so this is the list of the persistent winter halves that occurred in 2023. So we had 4 advisories and 3 watches throughout the state. And so lastly, I'll leave you with this slide. So for our lakeside homeowners, what can you do to kind of reduce your chances of having a halve occur in your lake? Reduce your nutrient inputs, so try to use phosphorus-free fertilizers on your lawn. Make sure you're doing proper care for your septic system. Get that pumped every 2 to 3 years. Make sure you try to manage any stormwater runoff. You can also add buffers to your shoreline to help stabilize, and that'll also try to help filter out those nutrients going into the lake. And be a responsible boat owner and pet waste management. Make sure you pick up after your pets. And of course, participate in any lake protection programs. So thank you so much for inviting me, and I hope you enjoyed the presentation. Do we have time for questions?
Yes, we do.
Okay. Okay.
You said that it can occur in tidal areas. Is that more inland where it's brackish? Does it exist? Can it exist in salinity?
So there are different types of algae that can occur in saltwater environments. So you'll see that more so in like Florida and things like that.
Like red tide?
Yeah, like red tide, exactly. So any, okay, gentleman in the hat.
Yeah, I have 2 questions.
Sure.
When you say the milligrams per liter, is that basically from ELISA or is it from the county?
Micrograms per liter. So that, when I was discussing that, that's from the toxin analysis.
But, okay, so but what does it come from? What is the toxin analysis then?
The toxin analysis is through the Elijah method. So that picture of that cube in there. So that's basically what that comes from, screening for. So the toxin comes from the cyanobacteria, but we're screening specifically for the toxin.
And the other question was,
Yeah.
you mentioned the cyatometer?
Yes, yeah, yep.
Is that what you actually count? Oh, no, when you count physically under a microscope, what do you count, density?
Yeah, so we count basically abundance overall, but we're only looking at cyanobacteria, right? There's other varieties of algae that can be in that sample, but we're only counting for that. So again, if you imagine a string of pearls, we're counting each individual circle, which is more time consuming and tedious, but that is the necessary procedure that we have to do. I hope that answered your question.
Great presentation.
Thank you. Okay, gentleman in the hat, other hat.
Since most representatives here are from volunteer associations,
Mm-hmm.
and I'm not a scientist, but to simplify for me,
Mm-hmm.
the toxins are released from the cells,
Mm-hmm. So,
and how does that occur?
yeah, so getting a little bit into the science here. So not all cyanobacteria can produce toxins, potentially. So the way to determine specifically what that is, which again DEP is investigating, is through metabarcoding. So looking for the specific genes in the cyanobacteria that produce the toxin. So that's why we just screen for the toxin currently, because we don't have that capability right now. But again, it's all potential. So when the cells start to break down, they can release the toxin into the water column.
And what normally causes that breakdown?
So it could be naturally through decomposition, it could be through potential treatment as well.
Okay.
So it's all, okay, sorry, we have a few people in the back waiting.
So you mentioned earlier the rapid response to a HAB alert. What is a rapid response? What does that look like? What do you do?
So when a HAB report comes in, it depends on where specifically that HAB is and which division it goes to. Again, typically it comes into our division. So we'll get the report, we'll say, okay, we have to go to, we'll just use Lake Mohawk for an example, Lake Mohawk. So one of our field biologists will go out there and we'll do an assessment to confirm that, again, because as you can see with some of the pictures, sometimes it can be difficult to tell if it is a HAB or if there's duckweed or water meal or whatnot. So they'll go out, they'll take some measurements with our phycocyanammeter and other equipment. They'll take a sample, they'll bring that sample back to the lab, and then that's where I come in and I start processing that sample and screening and doing counts and whatnot.
So it doesn't include a treatment or a solution, it's just an assessment?
Correct.
Gotcha.
Okay, sorry. Yep.
So I would like to understand the coordination of agencies with the individual lake communities. So if a lake uses one of those borrowed meters and detects a high count, and now the lake would, my question is, what does the lake do at that point? Do we notify the DEP or the local health department? And then my next question is, whoever we notify first, let's say hypothetically we notify the DEP, am I correct in assuming then the DEP comes out to verify it, and then they would notify the local health department, and then the local health department would tell that community to close their beaches? And if that's the scenario, when does that local lake community have the opportunity to test for a second time in hopes of finding a lower count and can reopen the beaches. I'm trying to understand the connection between the agencies and the timelines.
Okay, so connection between the agencies and timeline, I can't necessarily comment on that, but I can have somebody get back to you on that, because that's not necessarily specifically what I do. But with this meter, so if it's lent out to an association and they get a hit on, okay, potentially a watch number, you would report through that HAB reporting app, and then from there, our HAB coordinator, Chris Coons, who is our bureau chief, then will coordinate further on who needs to go out and assess and that sort of thing from there. Does that kind of answer your question, or do you?
I'm just trying to figure out who, so once it's determined it's a high account, who advises that lake community what to do? To close the beach or not close the beach? To put up what particular signs? Who does that? Where does, it seems like you're talking about measuring the count, but then what happens after that?
Yeah, so again,
I'm really concerned also for the opportunity to do another count so that the beach is not unnecessarily closed for a long period of time when the count dissipates.
So from my general understanding is that then Chris Coons would then say, okay, we're going to send a biologist out. Biologist comes out, just as I had mentioned to the other question earlier of that similar process. And then from my understanding, DEP probably then communicates with DOH, but again, that is not my necessarily responsibilities as far as knowing that process. So that is something that Chris Coons can get back to you on that.
Yes. The meter, that would be on loan for the season, is that how that works?
Yes.
And, well, I'm from White Meadow Lake, he's our site manager, I got my lake chair is also in the audience. We'd be interested in finding out about getting one of them.
Okay.
And, how is the best way to test with that? What location, do you go out waist deep? Do you take a few samples and then sample that cup of water? Opposed to just going to the shoreline and scooping up?
Yeah, so you would go out into the water. There's a line on here that says submerging line. You would take 3 readings, and then you would take the average of that, and then again report through our HAB reporting app. So if you email that email that I had on the other slide, I can pull it up. Hold on 1 second.
So that would be at the shoreline or out in the water?
At the shoreline.
At the shoreline?
Yeah.
You would take that?
Yes. You can take it out in deeper water if you would like as well. But again, just note that in the comments when you do make your report.
Okay, and you mentioned about cleaning up pet waste and stuff. Every lake community, except for one who conquered the challenge, has a major deep goose problem.
Yeah.
And it seems like there's such an expense and barrier from the government to control these pests, and that's what they are. They're not endangered. There's millions of them. And all they do is eat and pollute. But why can't your agency and other agencies, lobby the DEP or who's at Fish and Wildlife to make something where you don't have to hire some department to hopefully capture them? It costs lake communities thousands of dollars, and it's a gamble because someone scares the geese away. And idling the eggs is a very minor help, but it's a major issue with the water quality, and it's causing major problems now. And it seems like there's nothing coming to assist to control these pests. If it was some other kind of animal, they would have a hunting season.
Yeah, that would fall under Division of Fish and Wildlife. I encourage you to reach out to them and ask that.
And maybe Paula should do that.
Okay, all the way in the back, Bill.
So you had mentioned responsible boating. So I know, a number of years ago, there's a big thing with our lake. It's like don't attract hitchhikers. It's all about zebra mussels.
Yep.
Is it the same kind of thing with if somebody takes a boat to say pick on them, but I know they had a big outbreak a few years ago.
Mhm.
If they brought their boat in without washing the trailer or the boat and all that stuff, can they transport that
Yes.
stuff into the lake?
Yes, potentially.
So it's the same thing, wash your boat before you, yeah, okay.
Yes, 100%. My background is in invasive species management. Invasive species, please, please, please clean your boat. You don't want invasive aquatic pests. So far, we don't have zebra mussels in New Jersey, knock on wood. But if we end up having zebra mussels, we've got lots of big problems. Okay. Lady in the green.
Sure, thank you.
I'm sorry.
Very nice, thank you. So Upper Greenwood Lake has a swamp area where water comes in.
Uh-huh.
We tested there with the meter last year.
Mhm.
But the beach is 2 miles away. So what's the scenario of having a high count maybe at the swamp area, which you could expect?
Mhm.
But the beach is okay. Is that going to be a problem for the operation of the beach?
So again, with HABs, they can move around. Sometimes they only stay in concentrated areas. Sometimes they don't. So there's potential for downstream toxin transport. So that's something that's still being researched currently. USGS is doing some research with that.
But would you expect the beach to be closed if you have a high count 2 miles away?
Potentially, but it's possible that it could not show up as high counts in that area either.
Okay. Thank you.
Yeah.
Yeah, when you get a grab sample in to when you get a ELISA analysis out, how long does that take?
So the ELISA test, again, sometimes it depends on how many samples we're running at a time, but usually only within a couple hours. Okay, sorry, questions over there? Yes?
So it sounds like there's no procedure. You have lakes that are 500 acres, you have lakes that are 200 acres, lakes that are more than 500 acres. When you sample with the meter, and I think people are getting to this, is there any kind of procedure the state has on how many samples you collect to really assess the issue in the lake? And I know it's a constant thing. If you have a little bit in a swamp area, that's kind of like, oh, now I got to keep an eye on that.
Mm-hmm.
But that doesn't necessarily say shut down my beach today.
Mm-hmm.
It might be, oh, I got to keep an eye on that. I got to test every day.
Mm-hmm.
But I think that's the general fear I think everybody has is once you shut down the beach, by the time you get the analysis and stuff back, it's usually a couple of weeks, and you're losing 25% of your summer.
Yeah.
You have a lot of angry residents.
Oh, trust me, I know. So again, our response strategy is more reactive than, doing kind of preventative or preliminary monitoring. We do have a lakes program that does go out and do screening all year long, but it's based on a probabilistic model. So, not every lake in New Jersey is getting visited every year because we kind of are on a rotation of that. So I can also go ahead.
It seems like there should be a kind of like a sampling manual based on the size of your lake, at least some protocol.
Yeah.
Because, you get these little patches, people call up on our lake, oh, I got this little patch. It probably is cyanobacteria. It's like 3 feet by 4 feet. If I sample in that area, I'm probably going to get it, but it's a 500-acre lake.
Right.
Like,
not
is that
one
a
lake
reason,
is the same as the others too.
Is
right.
that the reason to shut down our lake? And my suggestion is no, it's not. It's a wow, I have to watch that, and I have to do some kind of treatment, alum or something, but it's not shut down the whole lake.
Yeah,
so
And
I guess we're looking for the state to come up with some kind of guidance that would give you, it's all or nothing right now.
Yeah, and again, our concern is public health. So, if we were to measure and get results of, okay, we've got a HAB, but maybe we don't see that the HAB isn't across the lake kind of thing or covering the entire lake. Again, That's kind of like our focus right now, but I understand your concerns and where you're coming from with that. Again, there may be more information on sampling in that lake management guidance that the HAB expert team put together, so I encourage you to take a look at that as well. Okay.
So you mentioned at the beginning that harmful algae has been around for 30 billion years or whatever.
Mm-hmm.
We started testing in 2019. I've been on the lake 38 years. I thought that was a long time. But when I had kids in the water, now I have grandkids in the water. You take a shower when you get out. Keep the dogs away. Keep the little kids away. Don't ingest the water. Don't eat the fish, and that's it. And now we're going into this realm of beach closings, which I'm all for protection, but it seems it might be overkill in the long haul.
Okay. Thank you for your comment. Okay, in the
So I'd
back.
like to follow up the last two.
Yeah.
My concern is, I'm concerned about the health aspect, but I'm just concerned that we may be shutting our beaches down for a longer period of time than is necessary. So I'm asking, what is the DEP going to do this summer? Because the summer is only 8 weekends. What is the DEP going to do to get back to us for that second reading, so that if the problem dissipated, we can open up the beach again? Because I get, last summer I felt like closing our beach for longer periods of time than we needed to do. So is the DEP aware of that? And can they help the communities by responding quickly to the second testing question?
Again, I will have our HAB coordinator reach out to you. If you can give me your email address, he can answer some of these questions further for you.
I wasn't the question I was asking. I'm asking, can the DEP please do something about it?
Okay. Thank
Question
you.
over here, Emily?
Yeah.
Is your presentation available?
Will it be available?
Yeah, we'll post
okay,
it
all right.
online on our website.
All right.
Give us a couple days.
I figured that would be the case, but I just wanted to.
Question?
What about, at what point do you suggest that we treat? If we come up and it's 20, do you suggest that we start treating the lake so that, to get rid of it, or, and does the DEP assist with any of that?
I can't necessarily comment on that because that's not, I'll have the HAB coordinator reach out to you about that information because it's fine.
Yeah, can I ask that, to build on that and say, what treatment is there for an outbreak? I did not know that there was any treatment more than prevention treatment.
So there are a variety, so my background is in consulting. So there are a variety of different treatments that can be done to try to control the algae in general. There are copper sulfate treatments, there's alum treatments, things like that. So there are.
Those are things that you do, that's part of the normal treatment of your lake, right? It's not, okay. We do that, so.
Yeah.
If people have more in-depth questions, can they email the questions to that email address and have them answered?
Eventually they can get there, yes.
Okay.
Yes.
Also, you mentioned a webinar. Is
The
that
answer
on
is
one
yes,
of
but
your sites?
how long it's
Does
going
it
to
go
take for
beyond the
response
topic
is
of
the
today?
So the webinar that I mentioned was from Tuesday. So that was a lake management guidance session. I'm hoping that more of those types of webinars are going to come out with various topics.
That webinar is on YouTube.
What's that?
That webinar from Tuesday is on YouTube.
Yes. Yep. It's available on our website and on YouTube as well. So if you,
YouTube under.
I'm sorry, what was that?
On YouTube under DEP HAB.
I believe under DEP HAB, but if you go to the NJDEP website under our HAB tab, you'll see that there's a link there as well. There's one question, one more question.
Okay, quick question.
Yeah.
We found particularly last summer that our phycocyanin and cell count correlation was off from previous summers. So actually two questions. One is, was that experienced statewide, that there was kind of a change in correlation? The second is, does DEP consider the cell size when they make the general guidelines about cell counts? Because we find sometimes during the summer, we get a strand of cyanobacteria that's got such a tiny cell that our count shoots up, even though our water conditions don't appear to be bad, so to speak. And so that throws us off. So we might, you know, think leaf closer when in fact the water looks fine.
So when we do our identification and enumeration, we aren't necessarily doing measurements, again, because we have lots and lots of samples coming in, so time is of the essence to do our counts, so we don't necessarily do measurements with that. As far as the measurements for phycocyanin, you were saying were varying?
I'm saying our correlation.
Or correlation.
From previous years.
Yes, yeah, so that graph from earlier, when we did the reevaluation, we did see some changes with that as well across the board, so.
And did you speculate as to why?
So there could be some variation happening based on weather patterns, because phycocyanin and temperature are correlated, so that could explain that. Okay. One question?
Okay, I'd like to actually piggyback on that last comment, because we also experienced that where the cell count particularly, and I forget the name of, and I probably couldn't pronounce it anyway, but
it's okay.
It's really small, and it's just from a practicality standpoint, wouldn't it be a benefit to do the analysis with regard to the propensity for the toxin generation across the various types of cyanobacteria, so that we would be then better served as far as, you know, if we've got a hit for, you know, say 30, but it's associated with these very small cells. And so again, the risk is smaller than if we get the ones that are very large, and we have a hit of say 15. So, you know, that's where.
Right. And as I mentioned earlier, again, certain cyanobacteria have the particular genes that can produce the toxin as well, so that's kind of where we're potentially moving into that as a benefit for screening for that potentially.
When is that available?
Huh?
How far is that research?
To be determined.
Okay, all right.
So we're still in discussion of it. So.
Okay. All right. Thanks.
Yeah. All right. Thank you so much, everybody.